For today's PowerPoint, we will be learning about gel electrophoresis. Now, what is gel electrophoresis used for? Gel electrophoresis sorts DNA strands according to lengths or proteins according to charge. It is used for DNA fingerprinting to compare the DNA between several organisms, isolating DNA fragments, isolating proteins, and various other uses. The components of gel electrophoresis include a plastic shell, a buffer solution, agarose gel, and a power supply with wires. The plastic shell is there to hold the gel and DNA. The agarose gel is used as a filter to sort the DNA strands according to length. The power supply and wires provide the electric current needed to pull the strands apart based on strand lengths. Now, how does it work? Well, gel electrophoresis separates DNA fragments by applying an electric field to move the negatively charged molecules through a matrix of agarose. This separates strands according to length, with the heaviest strands or longest strands being near the top near the insertion site, and the shortest or lightest DNA fragments at the bottom of the gel near the end. Also, if you look to the image given below, which depicts a basic gel electrophoresis lab using 3 different restriction enzymes that digested DNA plasmids, you can see a thick band. This thick band just tells you that there is a high density of strands of the same length, okay, in that one area. Now, how do I use gel electrophoresis? The first step is to place the gel in the apparatus. The gels can be bought online, or you can make one for yourself using the plastic shell of the gel electrophoresis apparatus as a mold. Once you have the gel in the apparatus, you can submerge the gel in a buffer, but do not completely cover the gel. Then you can choose the DNA that you want to compare, cut the desired DNA with enzymes, and dye the DNA. It is important that you dye the DNA so that you can see it in the gel or on the special gel electrophoresis paper that you can dye. Now, once you have cut your desired DNA and dyed it, you then have to place the DNA in a slot in the gel. The first slot of the gel is usually reserved for a marker, okay? This is a specific sequence of genes that have already been isolated and identified that can then be compared with other organisms, okay? The second slot can be used for another organism. Then you can place comparing DNA of different organisms in the subsequent slots. All right. Once you have finished doing that, and you must be careful when placing the DNA in their respective slots using a specialized scientific pipette that is measurable to not disturb the gel or cut it, as this may result in skewed data. Okay, now after you have placed the DNA in the gel, you can attach the positive and negative wires to the respective areas on the power supply and gel electrophoresis apparatus, then turn it on, in which you need to wait a while. As you watch the strands move to the end of the gel, you need to turn off the power supply an inch away from the end of the gel, as to not let any DNA run off of the gel to the other end, as it will not be comparable if it is not on the gel. Once you have turned off the power supply and let all of the fragments in the gel settle for a while, you can either use gel electrophoresis paper to color the paper and use that to compare the strands of the DNA, or you can look directly at the gel. On the next slide, you will see an image of a gel that has already been run in its plastic container. Okay. As you can see, it is slightly hard to see in the gel, and I recommend using a gel electrophoresis paper to dye with the bands and see on that paper. You can then make photocopies of this same piece of paper and hand out to fellow scientists, use in research papers, or with students that you may be teaching. Now notice that the strands or DNA fragment bands on the black image to the right are all separated nicely. There are several strands present in this image, which may be more difficult, but is not impossible. If one strand or one band on a column is at the same height as another band on another column, you can say that they are of the same length. Okay? And if you used all of the same restriction enzymes to cut the DNA, you may be able to say that they represent the same gene. Say, brown hair in somebody, or many other genes. Now, the gel electrophoresis has many uses within genetic engineering. Within genetic engineering, scientists use gel electrophoresis to isolate a desired gene to insert in another organism, to isolate a desired piece of DNA to clone, in which you would insert the gene into the plasmid of a bacteria and allow the bacteria to then reproduce and clone that specific gene that you want, that then needs to be isolated again. You can also use it to compare the genetics of organisms before and after you have genetically modified them, along with many more uses. Now, the lab activity given on the last slide of this presentation gives a website to a fun, interactive virtual gel electrophoresis lab activity that you may present to your students if teaching a class about genetic engineering and gel electrophoresis. It goes through all of these steps of gel electrophoresis with animated images that you can see, along with everything that may be going on, critical thinking questions, and all of the materials needed for an actual lab. This may be more advantageous as gel electrophoresis can run on the high end if you're doing it for several students or each student in your class, and a virtual lab may be more cost effective. Now I hope you go onto the website, see the virtual lab for yourself, and enjoy. Thank you.